LSM, I don't know, which means Shugai is the surname, I think. And the title is The Rise and Fall of Biophony and the Drivers of the Acoustic Space.
Hi, Johnny, and it's Larissa Sayuri Moreira Sugai. Don't know why it was LSM. So, can you see?
Just a moment. I don't see yet.
It's a black.
Yeah, we see it.
Okay. So, hi, and thanks for showing up. I'm Larissa, and I'll talk about the rise and fall of biophony and the drivers of the acoustic space, starring Larissa, Camille de Joquer, Thiago Sana Freire Silva, Tadeu Siqueira, and Diego Yusia. So we are all here, perhaps because we share a common interest in animal sounds, and which are central to the biophony component of soundscapes. And there we are, recording environmental or animal sounds because we want to do our best to extract meaningful biological information from biophonies and understand the ecology of animals. And at the most basic level, we can record sounds and determine the identity of species using manual or automatic analysis of their calls. And to extend the operation of this form of passive sampling, some pioneering recording systems were developed, but they are not that popular, and you may imagine why. And the release of new automatic sensors with scheduling functions to record at distinct periods with a given number of samples per hour, and distinct recording lanes, this caught the attention of ecologists. And we wanted to know how this technique was being applied in ecological research in terrestrial systems. And we made a kind of a dossier based on a literature review of 460 articles published until September 2018. Well, we can see the first articles on this topic appear in the 90s, and a discrete expansion up to 2010 with the use of automatic devices appearing more consistently. They're represented by the gray bars, and an exponential increase after 2010, especially promoted by the use of these automatic sensors. The first works, and the majority, were interested in activity patterns, habitat use, the efficacy of the method, and applications to biodiversity assessments. And you can see more diverse topics being addressed after 2000, and the largest concentration of articles on all topics after 2010. The largest portion of articles was related to bats, followed by birds, anurus, soundscapes, no-volume mammals, and invertebrates. Notice that the use of automatic sensors, the white bars on the right and the continuous lines on the left, was associated with most of the articles for all groups except bats, and the majority of studies were concentrated close to 2018. And very likely this trend has increased in the past 2.5 years. Most field work were concentrated in the global north, and we can see important contributions of some pioneering research groups in the USA, France, Australia, and Costa Rica. Considering the survey designs, the daily period of study was associated with periods of higher probability of activity. So you see that bats and amphibians focus on night and dusk, and the birds and soundscapes were mainly investigated on the dawn and morning periods. With temporal sampling, most studies focus on specific dial periods with continuous recordings, and fewer investigated full 24-hour scans. And in the studies using discontinuous recordings, most use 1 recording per hour, and from those, almost half use recording lengths lasting between 10 and 30 minutes, and the other half using less than 3 minutes. And although we saw that some recording schedules were more frequent than others, a variety of combinations are possible, and their adequacy will depend on the goals, the focal organism, and on the budget, of course. So a suggestion would be to have an adaptive sampling procedure, And first, obtain estimates for 24-hour recordings, for instance, here in this example, species richness. And then, by subsetting distinct schedules and comparing them with full 24-hour estimates, you can obtain the efficacy of the sample effort and weight it by the associated costs. And then the best solution will be somewhere between how much information is represented with the sample effort and the feasibility to apply such effort. Okay, so we just saw that acoustic monitoring can be applied for many purposes in ecology and conservation, and that standardized data collection with the benefits of high temporal resolution enabled to address interesting questions in animal ecology. So going back to what biophonies can tell us, we saw that a lot of research focused on activity patterns of which mostly details diel and seasonal activity of single species and its abiotic correlates. But when we scale up to entire ecological communities, the approaches often sum up all species recorded at a site, disregard of the timing that distinct species were active. However, we do know that short-term variation in species activity is widespread in nature. For instance, consider these annual assemblies in the beginning, the mid, and late night periods. So it remains to understand the influence of short-term dynamics in the structuring of ecological communities. And in this sense, we investigated short-term dynamics of annual assemblies in the Pantanal wetlands of Brazil using acoustic monitoring in 39 sites that represented gradients of a vegetation heterogeneity with both in situ measures and a remote sensing vegetation index, and the ecological context with a metric representing the phylogenetic dominance of the Anurian families, the SOS score there, and species richness. So, given the composition of the active species on each night period, we assess the pairwise similarity of species compositions between night periods. And we did that using two temporal resolutions, the 1 hour resolution and 3 hour resolution. So, and we found meaningful differences in the compositional similarity that was attributed to a night period only for the early and late night periods. That for both resolutions. And here in this two-dimensional representation of species composition in the early, which is the red dot, and the green, in the late with the green, for each assemblage, we can see a variety of changing forms between night periods. So we then investigated the ecological correlates of activity of these assemblages. Then we found a positive relationship with the phylogenetic dominance of assemblages, where an increase in similarity between the early and late night periods was associated with an increase in the dominance of species from the Elide family, here on the right. And then a decay in the similarity was associated with dominance of species from the Leptodactylidae family, here on the left. And this was only detected when we used the 1 hour resolution. We also found an association with an axis representing habitat heterogeneity, where an increase in the habitat represented an increase in the density of vegetation of the terrestrial and aquatic portions of the habitats. And then what we see is an increase in similarity between night periods associated with insights with more heterogeneous habitats. So in the light of resource partitioning, in assemblages dominated by Leptodactylidians, in the left, they are terrestrial and have higher encounter chances, and also in low heterogeneous sites. Species will be more susceptible to direct negative interactions, while in assemblages dominated by Elidians, they are arboreal species able to explore the vertical stratum, and in more heterogeneous sites, species would have more chances to segregate in space. So, a higher compositional similarity at sites with larger conditions for spatial partitioning and lower similarities in sites with higher competitive potential for space could represent an interplay between the habitat and fine temporal partitioning, which could reduce the effect of negative interactions and contribute in promoting the stable coexistence of species. Okay, so in addition to the idea of diameter, acoustic signals are traits that are subject to a number of selective pressures, such as sexual selection, masculine interference, attenuation, degradation through the environment, and predation pressures. Some of these pressures would influence the acoustic space of communities, here represented by these two axes and the colors. Most notably, the acoustic niche hypothesis predicts that selection against interference would promote a segregated acoustic space. And the acoustic adaptation hypothesis predicts that selection for optimized transmission would produce an aggregated acoustic space. However, the distribution of traits in ecological communities, including the acoustic traits, is also subject to other forces, such as phylogenetic niche conservatism, allometric constraints, and processes acting on the assembly of communities. For instance, if environmental filtering or competition are important forces in community assembly, and the ecological traits involved in this process are correlated with the acoustic traits, and both are phylogenetic conserved, a possibility is that the acoustic space would be a side effect of the interplay between these processes. Additionally, the acoustic space can also reflect the assembly of communities relying upon social information use, also known as extended communication networks. So basically, it's virtually impossible to infer the process behind the acoustic space only by looking at the pattern alone, because multiple processes can drive similar patterns. So the pattern should be carefully interpreted, and this is perhaps the main take-home message. So we tackle the drivers of the acoustic space of the Anuran assemblages in the Pantanal Atlantis. And we first assessed the general acoustic structure. And we did that in a similar way that was explained by Slade Alien-Nakins in his talk. We computed the mean distance among Cosainale species. Then we randomized species and created new distributions of such distances and summarized them with a standardized effect size, where higher observed distance would have positive values and lower observed distance would have negative values. So we found that the acoustic space of such neural assemblages tended to be more aggregated than expected by chance, even when you control for the effects of body size and phylogeny. And these results were especially meaningful for peak frequency and spectral evenness. Then we plug in the ecological gradients of environmental structure and the ecological context, and the ecological context we represented with the phylogenetic similarities as a proxy of ecological resemblance and body size. And we plug in all of this using structural equation models to control for the effects of the environment on the ecological context, and which also enable us to extract both the direct and indirect influences on the acoustic structuring of the assemblages. And we found a positive relationship between phylogenetic similarity and peak frequency similarity. We found that the environment influenced the structure of body size in assemblages, which in turn were negatively associated with peak frequency similarity. The structure of chaos timing was poorly explained by the predictors, and spectral evenness similarity was positively associated with vegetation heterogeneity. Now, piecing all together. First, the overall aggregated acoustic space contends against the acoustic niche hypothesis. The indirect effects of environmental structure on body size and phylogenetic similarities were negligible, indicating no indirect influence of environmental filtering. Acoustic and body size had no strong phylogenetic signal. So the positive relationship between phylogenetic and peak frequency similarity should be considered cautiously, while the negative relationship with body size similarity is actually opposite to what would be expected upon the niche conservatism. Then upon the acoustic adaptation hypothesis, an increase in vegetation heterogeneity would pose constraints in signal transmission and should presumably be associated with aggregation on the acoustic space. But we found exactly the opposite. So we also rule this out from the potential drivers. So we finally argue that our findings can be related in part with phylogenetic niche conservatism, but we also raise up the possibility that social information use could underlie the relationships between acoustic, phylogenetic, and body size, particularly given the theoretical benefits of information use, with the assembly of species with similar ecological requirements, but reduced ecological overlap considering body size. So in conclusion, although we might be tempted to consider biophonies as deterministic outputs generated by the acoustic niche or the acoustic adaptation hypothesis, these are still hypotheses among other potential hypotheses that should be tested, and when tested, should ideally consider the ecological and the behavioral context experienced by communities. Well, with that, I'd like to thank you all for your attention.
Thank you, Larissa, for your great presentation. And I suppose that many have to make questions, but I also have to say that your presentation is very, very complex, and you presented a huge amount of data and information and thinkings. So I think there is a lot to discuss about your work. And first, my compliments for your work, including the first part that was a very interesting review of the bibliography. And it is really important to have such an analytic view on the bibliography to understand where to go and how to manage new directions in the research. Any questions? Okay, Bernie? Bernie, switch on the microphone.
Yeah, okay. I thought your presentation, Larissa, was fabulous. It was very, very interesting and very compelling. My question to you is, when you're doing the recordings and gathering the data at these various sites, do you take into account the actual limitations of the habitat itself? In other words, when we were doing mapping in Costa Rica, we found that the different sites that we were recording at were defined kind of by amoeba-like shapes rather than the 100-meter shapes that a square meter that they had all created at these research sites. And so we found that the various microhabitats that we were recording needed to be defined very differently. And we laid over a map of those things so that we could get those structures adequately defined. Did you do the same thing when you were gathering your data?
Yeah, thanks, Bernie, for your compliments and for your question. So if I understood, like, you mean that the environments have such a complexity that sometimes if we just reach the places and try to sample things, we can't really represent all of that complexity, right?
Yeah,
so
sure.
First thing I did when I was designing the study, I mapped the region using both optical and radar images. Mm-hmm. And then I created some mappings and also use some indexes of vegetation, some continuous measures, mm-hmm. to represent, and then based on that, all of that variety of environments, I first look where can I reach the places by the road. So, which is really a limit. So mm-hmm. we tried to do our best to represent from everything we had distant points, 39 points among all the gradients of the macro, which were buffers of 200 meters. And that's what we try to do to make sure that all potential forms of species from Anurans could be represented in this study region.
So did you take into account the topography and also the vegetation and the habitat as well?
Yeah. Oh, the topography. I mean, this region is a lowland, and it has only like for really hundreds and hundreds of kilometers, such variation of centimeters. But since it was kind of a more not big spatial scale, the topographical aspect we didn't consider. But yeah, the kind of vegetation was totally taken into account when we stratified the sample points.
Okay, because we noticed when we were working in the desert, Southwest desert, North American Southwest, we noticed that when there were altitude changes of sometimes 2 or 3 meters, we got a very different result. And also, my other question to you would be, how do you account then for the acoustic niches that we find in habitats that you even showed on the screen? I mean, how do you account for that? And we found that almost in every tropical site we recorded.
So the other sites that we didn't represent, sorry, I think I couldn't get it.
I'm sorry?
I'm sorry, I couldn't understand which other sites.
Okay, we've recorded in many tropical and subtropical sites around the world. And in each one of them, they're validated in some way by very distinctive acoustic niches. How do you account for that? I mean, it's there in the spectrogram.
Yeah, well, it's there in the spectrogram, but there are many things that happen during the historical course of species, like distinct kind of interactions that occur that went like a pressure to somewhere in sino evolution, or even historical aspects that changed the structure of communities in there, and so many other potential things. Sometimes we also term like the ghosts of past competition or the past evolution that we can't say about it just by looking at the pattern of distinction of acoustic niches.
What about hearing it?
Yeah, I understand this is really, it's really there, right? I know what it's about.
I hear it. I mean, I hear those discriminations between species, between the ways that they're arranged in a spectrogram, because my background is in music. Yeah, and so I listen to things musically and structurally.
Yeah, you know, I also see it really distinct, but I can't really say that it's a past process of competition or something.
Yeah, it may not be.
I mean, that the pattern is for sure. You can describe it in any theory, but we can't really say about the process itself. That's my point. Just by using the pattern, you know. And sometimes we are talking like all the spectrogram, right? Including mammals and invertebrates and neurons and birds. And my feeling is that sometimes we have to take some zooming and to see what's happening with biological entities that share common ecological requirements, like some guild of, as I studied, Anuran assemblages, or some bird communities, to try to understand this more fine ecological and evolutionary process.
Yeah, and you may be right. I mean, we don't know enough about this field yet to be able to make those kinds of, compelling arguments that are going to stand for the rest of life, because we're just exploring it now. Yeah. But I mean, I thought your presentation was great, and you really have some good rationale behind it. I'm not sure that it's like the great Paul Shepard, who was a naturalist in the U.S. who died in 1996. He wrote The Others: How Animals Made Us Human, and in that particular piece, he says, we human beings probably didn't evolve to understand everything that the natural world has to throw at us. So I'm with you. I think that that's really a good challenge, and it's something that we have to explore. Thank you.
Thank you, Bernie, and please come visit the Pantanal someday. Let's make some recordings in there.
Yeah, okay. I will.
Okay. Thank you very much to Larissa and to all the people making questions and the previous speakers, of course. And I let Almo to conclude the afternoon session.
I declare concluded the afternoon session because I think that there are so many hours that the people is in front of the screen, and it's time for something else. Anyway, thank you very much, and see you tomorrow morning at 8:30. There are no changes in the program. And David, can you have to say something about? No? Fine. Thank you for your attention, and see you tomorrow for the final day of this amazing Congress. Thank you very much. And open your video and make salutation. Bye-bye.
We thank you.
See you tomorrow morning.
See you tomorrow. Ciao a tutti.
Ciao a tutti.